Okay, welcome back, everybody, to our second presentation. Our next talk will be given by Dr. Greg Wu of Washington University, St. Louis Department of Neurology. And we had some good discussion in the last talk, and I'm sure we're going to have even better discussion for this talk. Please take it away, Dr. Wu.
Okay, thank you so much. I'm very happy to be here, and I'd like to delve right in since I have plenty of slides and lots to talk about. So these are my disclosures. I don't believe there are any conflicts, but if you do wish to address them during the talk, please stop me and I'd be happy to. I'd like to just give a brief overview. I think there was plenty covered in the first talk, and really focus on adaptive immunity and multiple sclerosis. I'd like to just highlight some of the current therapies as an example of what we do currently in terms of treatment for our patients with multiple sclerosis, before transitioning to highlighting briefly a few emerging therapies, some of which I think are very interesting from the perspective of spinal fluid examination. So this is Charcot, who we would, I think, attribute as the father of MS. He was a brilliant clinician, and at that time was really responsible for tying pathology to clinical disorders. And he was very keen to identify patients who had what he referred to as the triad of nystagmus, tremor, and dysarthria, and identified at their death features of pathology that we now acknowledge as multiple sclerosis. Well, what are those features? They're damage primarily to the white matter, loss of myelin, and sclerosis or hardening refers to the idea that these plaques or areas of injury, if you were to reach out and touch them, actually have a different consistency than the normal brain surrounding it, or the relatively normal brain. Not as spongy, hardened. And if we zoom in on those areas of sclerosis, we're able to identify using microscopy techniques that those territories are consistent of inflammatory infiltrates. The arrows in the top upper left here, just indicative of blood vessels that are engorged with extravasating leukocytes, which are seen at higher power here. These lymphocytes or leukocytes, white blood cells, are coming in primarily through the postcapillary venules. And in those regions in which those inflammatory infiltrates are present, there's a loss of myelin in the areas that are highlighted here with a myelin stain shown in blue. Well, what are the other factors that make us suspicious that the immune system's the culprit in multiple sclerosis? One is spinal fluid, and as I said, I will come back to this at the end. CSF or cerebrospinal fluid in MS patients typically have an abnormal elevation of white blood cells. That's the WBC here. Typically, a majority of those will be lymphocytes, sort of denoted here by L. At Washington University in St. Louis, we colloquially refer to the additional non-routine studies on spinal fluid as Trotter studies, named after John Trotter, who ran the multiple sclerosis section for many, many years, and performed these studies in his own laboratory before they became sort of more regimented and CLIA certified, et cetera. And these studies consist of identifying abnormalities in the antibody and protein content here. Actually, it's very worth mentioning that spinal fluid should have certain characteristics, but there is such a heterogeneous nature to multiple sclerosis that one thing that we really have to remember is that we should make this a disease of exclusion. We're trying to point to MS, and it's very challenging because there are no diagnostic tools that are absolute. There's no really one diagnostic test. If we could capitalize on the genetics, as was emphasized in the first talk, maybe we would have a diagnostic tool that would be highly sensitive and highly specific. But for the moment, we rely on the fact that we can't diagnose this disease without excluding other diagnoses first. John Trotter, really basically did the electrophoresis in his lab, taking serum, denoted here by S, and CSF by C, and running them in parallel, and asking whether or not there were protein contents within the CSF that were not present in the serum. Here we can see just an overall electrophoretic gel showing lots and lots of proteins that sort of segregate out, and you sort of squint and appreciate that there may be extra protein bands shown in the cerebrospinal fluid, not in the serum. When you increase the ability to recognize these antibodies by using isoelectric focusing paired with immunofixation, the bands present in the CSF of this MS individual shown on the right side of this gel really become much more conspicuous. These antibodies are one element to which we would point to the immune system as being the culprit in multiple sclerosis. Another commonly referred to piece of evidence to suggest that the immune system really is directly involved and may be responsible for multiple sclerosis is the experimental model commonly used to investigate the pathophysiology of multiple sclerosis called EAE, or experimental autoimmune encephalomyelitis. This is a disease that can be induced in any variety of different animals. In my laboratory, we immunize black 6 mice to some myelin component. In general, it's myelin oligodendrocyte glycoprotein, which in the right context with the right adjuvants will lead to disability that's observable in these animals as hind limb paralysis. If you take mice that have been immunized to induce this form of active EAE and culture immune cells in a dish with additional myelin components, such as the myelin oligodendrocyte glycoprotein and other inflammatory cytokines and products, you can then transfer CD4 T cells from this dish into a naive host and elicit the same disease that was seen from active immunization. So it's with this model that we have really pinpointed the CD4 T cell, a lymphocyte subset that can be sufficient and is required to induce this disease of EAE. So I kind of view the disease pathophysiology in a very rudimentary way that's shown on this slide, where that CD4 T cell needs to become activated. It has to be able to be directed toward a very specific immune target, and that activation takes place in the context of MHC class 2, which was highlighted in the first talk, borne by this antigen-presenting cell. We likely think that this is occurring outside of the brain and spinal cord, but once that T cell, the CD4 T cell, becomes activated, it needs to traffic through the blood-brain barrier, recruit other leukocytes, engage again with another antigen-presenting cell that is presenting the same target, after which a whole cascade of inflammatory events unfolds, leading to that demyelinating plaque generating damage to the white matter in the central nervous system. Well, these lymphocytes are very specialized, and I'd like to emphasize with this slide that the T cell that we just focused on and B cells that make antibodies have receptors that dictate what they're able to recognize. And all of those lymphocytes are, expressing a receptor that tells it what it immunologically will be targeting. So if we imagine that in our body we have billions upon billions of different specificities toward various targets, that would allow us to fight off infections or heal after injury, or even target cancer, for example. So the receptor specificity of the T and B cells matters tremendously. Once they engage and are able to see that target, our immune system takes over in one part by exaggerating the response by inducing proliferation of a single clone. That is to say, a T cell that recognizes myelin oligodendrocyte glycoprotein, once presented and told to go after it, will proliferate and create many, many more of the same T cell receptor bearing T cell lymphocyte that will then be a clonal population or expanded population that will go forth and exert inflammatory or immune reactions. So this adaptive immunity slide will anchor me now toward describing some of the therapies that we use to employ to treat MS patients. And same kind of thing, except I reversed it here. I apologize. The B cell now is in the center and the T cell is off to the side. The same engagement with the receptors, targeting a particular antigen. But the B cell here expresses many different unique surface proteins that allow us to target them. One of the proteins that's highly specific for B cells is called CD20. And many, many years ago, scientists speculated that they could remove tumor cells that were B cells, so lymphoma, for example, using a monoclonal antibody that targets CD20. and it was highly effective. And so we could repurpose that monoclonal antibody that targets B cells to remove them in autoimmune diseases like multiple sclerosis. And so the sort of immunotherapy was born out of the ability to selectively and specifically target CD20, which would selectively and specifically eliminate B cells. And so this was employed by a group out of the University of California, San Francisco. This article comes from 2008. It's a phase 2 clinical trial demonstrating how potent B-cell depletion therapy is for multiple sclerosis using the antibody rituximab. And just to walk you through some of the data, the primary data looks like this. They're only using about 100 subjects, okay, 35 placebo and 69 rituximab treated individuals who they hope are very similar because they randomized them to these groups. They double blinded themselves and the patients and said, hey, are you the same? We want everybody to have the same features to start with. And for the most part, they are. They've got these relapses that take place or annualized relapse if they put that sort of impression over the 1-year time frame. This trial basically only lasted for 48 weeks. And they showed that people treated with rituximab did have a significant reduction in the relapse rate over 1 year. Pretty impressive because the trial was actually designed to measure MRI changes, not clinical changes. Well, what are the measurements we use? If we're doing a clinical trial, how do we tell if people are doing better or worse? Well, there are a lot of scales here. We can measure the number of relapses over a period of time because MS patients will have disability that comes and goes, and we can measure that. We can measure how much disability an MS patient has, and arguably there are many different scales like the MSFC or the EDSS that tell us how disabled somebody is. But a real quick and dirty is to count up the number of lesions that are present on an MRI scan as a biomarker or surrogate for how much damage is taking place in the central nervous system. And there are different ways to do MRIs, one of which employs a contrast agent called gadolinium. And if you see gadolinium, it means that the blood-brain barrier has been broken down. So people can go and ask how effective a therapy is based on how many of these enhancing lesions can be counted during the trial time frame. Well, this is the MRI outcome from the rituximab trial. Now, they didn't start at the same place, unfortunately, but nevertheless, it's very, very clear. If you even stood across the room and looked at these two lines, they differ very substantially. The rituximab treated group in blue just got quashed here. They went to 0. And the number of new lesions, same thing, bam, went to 0. Whereas the placebo group continued to have these lesions that, persisted over the approximately 1-year duration of the trial. This is highly statistically significant and highly rapid. The first time point that they did these scans was 4 weeks, but after 3 months, boom, these patients really had very little disease activity whatsoever. Highly impressive and very, very encouraging as a therapeutic tool. Remember, this is a phase 2 trial, though. This was not approved in 2008. This is just evidence that this drug works. It wasn't until 2017, however, that we had a drug that targeted CD20 to treat multiple sclerosis. Why? Basically because of money. Here's what happened. This I mentioned was a monoclonal antibody that was developed initially by IDEC. Biogen and IDEC actually merged in 2003, but at the time that it was first developed for a lymphoma treatment, IDEC used Genentech, a very, very big company, to help them develop it for lymphoma treatment. And it was approved for non-Hodgkin's lymphoma back in 1997. But because of the merger with Biogen and IDEC, when this study that I just told you about multiple sclerosis was taken underway, it was done with Biogen's support. In 2009, Genentech had a shotgun wedding with Roche. So now the idea that there is some form of treatment for MS that's based on CD20 fell into Roche's lap, who had already developed their own humanized version of rituximab. So the determination as to what to do after that article I just reported to you was dictated by Roche and their ability to develop a whole new antibody trial, not capitalize on what was already known in a phase 2 trial, but move forward with an entirely new set of experiments to demonstrate the efficacy of B-cell depletion therapy for multiple sclerosis. So that really set us back, and it was very sort of like, upsetting to the field of multiple sclerosis. Nevertheless, Same type of therapy. Ocrelizumab is just a different version of rituximab that targets CD20, and we can see very clearly that ocrelizumab treated MS patients have very little MRI changes, whereas the control, now not placebo, but a different version of a therapy that had been approved since 1993, as a comparator, did continue to have some form of MRI activity. Very effective medicine, and we use it regularly. In fact, it's very standard now to treat anybody with multiple sclerosis with B-cell depletion, mainly with ocrelizumab. Well, going back to the idea, however, about latency, B cells that are eliminated by ocrelizumab are one of the main reservoirs for EBV. We just heard about maybe EBV being a driver of multiple sclerosis. And I actually think that's, you know, legitimate. This is data from 2007, or a summary of data that was known in 2007. So, you know, 15 years ago, the data that actually, you know, substantiates this kind of idea is decades old. So there's no question that the idea how EBV could be contributing to MS has been around for many, many years. This is to indicate that if somebody has not been exposed or infected to EBV, which is extremely rare in adulthood, we only think that maybe 2 or 3% of us have never been exposed to it. But those individuals lucky enough to avoid EBV have a very low incidence of multiple sclerosis, almost zero. Those with early infection of EBV, that is in childhood, very early on, have a certain risk. But those with an EBV that have infectious mononucleosis, a very fulminant manifestation of EBV infection. Most of us who get exposed to EBV never have any clinical indication that we have had that infection. Those individuals actually have the highest risk of developing multiple sclerosis. And as was mentioned, there was a whole series new based on military recruits about, you know, millions and millions of people donate their blood when they join the military, and have provided a great epidemiologic substantiation for the fact that EBV is linked with MS. But biologically, then we can go back to those oligoclonal bands from the spinal fluid and say, what do those antibodies in the spinal fluid recognize? And a proportion of them are recognizing central nervous system targets. Glial cam, for example, an antibody pulled from an individual who was EBV positive with multiple sclerosis. They recognized EBNA, a viral protein, but that cross-reacts, that antibody cross-reacts with a glial protein as well. So the idea, I think, immunologically exists that the B cell could be contributing to multiple sclerosis as a result of EBV infection. Well, what does that mean for us? The field's very excited about this. Why? Because we believe that we can now latch onto EBV as an immune target and eliminate it as a potential source for dictating whether or not patients will develop MS eventually or not. So Moderna, for example, a big player in vaccines, has started human trials of an mRNA vaccine for individuals who are adults that already have MS. Maybe we can target those individuals who have B cells latently expressing EBV that drive some pathophysiology. But prevention with vaccines is really the goal. Never, ever getting this disease in the first place would be ideal. So a lot of vaccination trials now targeting EBV are underway or being devised as we speak. Going back to this idea that we should target lymphocytes, well, maybe there's some feature of them being autoimmune driven by EBV, for example, in the B cell. But what is it about these lymphocytes that really matter to disease? Well, it's their ability to become clonal. It's their ability to divide after they've seen their target. And so a repurposing of a drug that's called cladribine that inhibits proliferation of immune cells because of their sort of properties has been in the works for many, many years. This cladribine is used again in a cancer setting for, I believe, hairy cell leukemia, but it is administered parenterally. The idea about multiple sclerosis at some point was, hey, we need pills. We don't need shots or infusions. We need oral therapy. So cladribine administered orally can still have effects on the proliferation of CD4 and CD8 T cells, also B cells to be perfectly blunt. But the idea is that cladribine can substitute for adenosine and therefore inhibit proliferation because there's no DNA ability to divide with this dummy molecule cladribine in place. And it only affects these cells because an enzyme is required to convert it to its active form. And that enzyme is present highly in B and T lymphocytes, but not other immune cells. So when we give this pill orally, we're able to see, for example, that the number of relapses over a year goes down. and that the number of patients accumulating disability over time is reduced compared to those receiving placebo. So these are those outcome measures I just reported to as some of those things we latch on to determining whether or not these therapies are very viable or not. And it is. Cladribine works. We think that it works by reducing the number of proliferating lymphocytes. And the intention here is to really determine how much of that inhibition occurs and how much do you need in order to limit MS disease activity. So in these studies called Clarity, blood counts for lymphocytes were obtained periodically. And the medicine's given very weirdly. It's given in cycles. So you see an immediate drop in the lymphocyte count here, followed by some recovery, but then a drop again because the drug is administered after another year. So these drops are expected. They're intentional iatrogenic lymphopenia. And then over time, when we follow these patients, that number of lymphocytes doesn't quite reach the normal count, but comes back up into a very healthy range. We got involved here at Washington University because we don't really think that the peripheral blood lymphocytes are really where the action is. We think it's in the spinal fluid. Of course, all these immune cells in MS have to get through into the brain and cause damage. So we think that that's what's the key linchpin of understanding how cladribine and other medications work. So we partnered with the maker of this drug and asked, could we do some spinal fluid testing periodically through the study? And we profiled these immune cells, not just by absolute counts, but by a new technique called single cell RNA sequencing. And we can determine every individual dot that comes out of this single cell RNA sequencing as an individual cell that we can characterize by their gene expression. And we put them next to one another on a 2D plot by the similarity with each cell it shares with the other cells in this analysis. So the group of cells up here, for example, we believe are myeloid cells, and the group of cells here in this cluster are lymphoid cells. Why? Because their gene expression kind of groups them together in their identity. Well, what we determined over spinal fluid analysis using single cell sequencing in cladribine MS patients, treated MS patients, was that at 1 year, there's an emergent population. It's not that the types of immune cells go down, we actually see a new type of immune cell that comes up. If we zoom in on that population, it's actually made up of 3 separate clusters of cells. And again, the power of single cell transcriptional analysis is very cool, and it's very useful in this day and age to claim, which I won't substantiate to you all, but to claim that in fact, we think that these subsets of cells are regulatory. It's not that there's a dropout of cells that go away that are inflammatory as much as or equivalent to the idea that an emergent cell comes back when there's a repopulation of lymphocytes that might be beneficial to an MS patient and help quell disease and make the disease more tolerable. Okay, going back to this idea about what these immune cells are doing and how do we target them, a future drug on the horizon is called BTK inhibition, or a small molecule targeting of the BTK complex. Bruton's tyrosine kinase is a target for multiple sclerosis in the future simply because it involves the transduction of signals from the B cell receptor. If you genetically are missing BTK, you do not have B cells, they don't develop. But using a small molecule to target BTK helps limit how much signaling comes through the receptor. It just so happens that BTK is also expressed in myeloid cells like macrophages, so using a small molecule inhibitor could also impair or inhibit function that the myeloid populations of immune cells is undertaking. Well, because of the attraction of the mechanism here, and because there's still a lot to be gained in the therapeutic window for multiple sclerosis, there are 4 different compounds that are actively being trialed at the moment from 4 different companies, and they all have slightly different properties, including what site on BTK the small molecule binds, and whether or not it actually binds covalently, et cetera, et cetera. So we're very excited that there are still many companies interested in developing therapeutics for multiple sclerosis because there isn't an entirely wholly met need for the treatment simply because the patients are still experiencing inflammation and disability. I won't go through this. I think it's just a lot of numbers to suggest that these in phase 2 trials could work. Second, the penultimate example I'll give to you for potentially new therapies is, again, targeting these immune cells, but targeting their specificity. The medicines I've just told you about are very non-specific. Oh, we'll get rid of all B cells. Oh, we'll get rid of all proliferating lymphocytes. We'll get rid of things that are just using BTK. But what about the ones that we really care about? The antigen-specific immune cells, the ones that target and destroy based on what they recognize from their receptor. Going back to the EAE system, we know that the CD4 T cell that has specificity to MOG is sufficient and required. And this process is really, driven by the antigen presenting cell. In our lab, in collaboration with another lab, we decided to use that MHC class II antigen bound targeting system to engage with the disease responsible CD4 T cell and destroy it using a system called CAR, or chimeric antigen receptors, which are lethal immune cells. We're turning the immune cell on its head. This now is our effector cell rather than the autoimmune inducing CD4 T cell. It's the target cell. And we can reduce disease in this animal model system with the scale shown on the left. This is how weak the mice are. Over time after immunization, they get weak to this extent. And if you treat those mice, once they start to develop weakness with the CAR therapy, the disease severity is reduced. And the number of those cells that are specific to MOG that are ending up in the central nervous system is reduced as well. So we think that we have a nice, cool system to employ. Keep in mind, again, repurposing, these CAR T cells have already been used in the autoimmune sphere. They're being used to treat lupus or even a disease that looks a lot like multiple sclerosis called NMO spectrum disorder. But all of these are using the targeting mechanism non-specifically. They're going after B cells again, B cells that express these molecules like CD20 or CD19. So we think that maybe refining this and making it a lot more specific and selective by changing the way in which this recognition component of the CAR T cell is utilized might make autoimmune therapies for MS and beyond much, much better. Lastly, I'll just emphasize, we want to get away entirely from pharmaceuticals and drug therapies. I think it makes a lot of sense if we were to be able to push the reset button on our immune system. And many people have tried this already using stem cell therapy. In this case, stem cell refers to the idea that you can repopulate your immune system from your bone marrow, the hematopoietic bone marrow, hematopoietic stem cell system, that is to say. So the trial that's ongoing now is really one that we've used to identify whether using bone marrow transplantation is superior to any other therapy that's currently available. In other words, there have been many trials, small but informative trials using bone marrow transplant that suggest that yes, in fact, MS can be effectively treated, but there's never been a comparator control arm, and it hasn't been large enough. The trial has not been large enough to be impactful. Here we're using the autologous stem cell from the patient. They get their bone marrow wiped out. We put the stem cells back in, and these MS patients will be followed for up to 6 years in order for us to determine whether this therapy is helpful. And there's data to suggest that, in fact, it is helpful, even though the progression-free, that is to say, the people that you want to end up with no more disease, stay at the top here at number 1. Even though bone marrow transplant seems like it might not be so great, we do think there's a certain population in which bone marrow replacement could be useful, and that's the relapsing remitting MS patient who is very early in the disease course. Okay, I'll summarize here. I really want to emphasize how heterogeneous MS is. That might really mean that one therapy for one patient will not be effective for another patient. There are an abundance of disease-modifying therapies that are available currently. I just highlighted the one, the ocrelizumab, because it's very good and it works in a way we think we understand. But the other one, the cladribine, we're still trying to reverse engineer. In fact, many of the drugs that we have available for MS therapy were non-rationally designed. They're repurposed from a cancer therapy, for example, or they are trialed based on faulty rationale. We have to go back and really understand why and how these medicines truly work in order to then understand the pathophysiology of multiple sclerosis. And believe it or not, there's so much more to come. I just highlighted a few things off the top of my head, but there are so many other therapies that are emerging now. And there's a lot of science behind getting, these therapies that make me very excited to be in this field. Thank you very much. I'd be happy to answer any questions.
Thank you so much, Dr. Wu. Really appreciate your wonderful talk. And I would welcome any questions from anybody who'd like to chime in before I ask my questions. So anybody want to have a question for Dr. Wu?
Yeah, I had a question. Can you hear me?
Yes.
It's Brad Moffat here from the University of Melbourne. So what's your thinking about I guess the heterogeneous response to the different therapies in MS patients? Does it indicate like just that there's several different origins to MS in, the initial immunoreaction of these people, or is it different genetics, or what's your feeling?
Yeah, my answer is yes. I think it's a lot of components. One thing that I didn't touch on is really how impacted the central nervous system is by these immune responses. And clearly some people have a better resilience to CNS damage. That could be based on the location of the lesion, the size of the lesion, the extent of damage within the lesion, the ability of that lesion to remyelinate, the number of neurons that are remaining after an attack. So I think in the CNS, I've completely ignored the idea that there's a whole other side of the body that's trying to continue its job and do that well. And we all vary, I think, in our ability to withstand CNS injury. But on the immune side, I do think it's probably a variety of different triggers, subsets. I'm a big believer in the idea that MS is actually a compilation of different diseases which might stem from an abundance of, in one individual, T cell component, and the other it might be B cell or monocyte, innate driven. So I think in that case, when we see a fluctuation, or I should say a difference in response to different therapies, that very well could be the understanding of the basis for that diversity amongst MS patients. I'm not sure if I totally answered your question.
Yeah, no, I come from an imaging background. I'm just wondering whether we need, in the future, if there's multiple origins, multiple responses, do we need better biomarkers for understanding the origin to better tailor the treatment for these patients?
Absolutely. And I think that one example of that would be Bob Axtell at the Oklahoma Medical Research Foundation. When he worked with Larry Steinman, the developer of a drug called natalizumab, or one of the contributors to that FDA-approved therapy for multiple sclerosis, they were interested in interferons. Type 1 interferon therapy was the original therapy for disease modification of MS, but there are some people who respond very well to it, very few, but some who respond very well to interferon therapy. And then there are some that don't. They postulated that there's something that we could screen ahead of time, a biomarker to suggest who would respond best to therapy with an interferon. And that could be applied to any number of drugs. For example, the cladribine story I just said. Well, if we do a spinal tap on somebody and we say, this person already has that suppressive population in their spinal fluid, I don't think they're going to have to amplify very much more. I don't think cladribine will work very well for this individual. Or, there's a million different ways to skin that cat. And I totally agree with you that we need something pre-therapy to determine the future-facing therapeutic outcome for individuals with multiple sclerosis.
I was going to say that I think that early clinicians were sort of, the question is, are you splitting or are you lumping? And they were lumping a series of symptoms together to talk about multiple sclerosis and many other disorders. And the fact is that there may be a variety of genetic backgrounds, a variety of triggers, a variety of ways you actually have those symptoms in a patient. And I don't think we know enough yet. I think this about an awful lot of disorders that are adult onset and chronic in a large way. I don't think that there's been enough research really into these particular etiologies of disease with an ability to split out the groups as opposed to lumping everybody together and calling it all multiple sclerosis. So that's my thought as a forward-thinking geneticist.
Other questions, or I certainly support the concerns for splitting instead of lumping. I certainly support the concerns for biomarkers. Hopefully we'll talk a little more about that in the next session, beginning at 2 p.m. But let me ask one more question for Dr. Wu, if I may. Could you spend just a few minutes giving a little more introduction about how clinical neurologists approach a patient, and how, when, where, why to discuss with a patient which disease-modifying therapy to start with or to try, which clinical intervention to attempt in the current state of the art for current practice. How does a clinical neurologist approach that discussion with an MS patient?
That's a really great question, but it's very difficult to answer, I think, only because I have a certain practice and my colleagues practice differently. And I know that because we're not at an academic institution, we practice differently than practitioners in the community. And we have bucked the trend toward having a practice guideline, simply because there is still so much unknown about the therapies that are being employed. That question, I'm sorry, I don't mean to sound negative about it, but is the question that we get repeatedly from pharmaceutical companies, because they want to have a sort of a Chinese menu that's dictated about how things are practiced in a standard fashion, and it's just not appealing to the MS group because there's so much still unknown. But I'll say this. When someone's diagnosed, we really do pay close attention to several variables: their age, their comorbidities, their ability to afford medications, and then a million other things that would be practical in consideration, such as the number of lesions that the patient has on their scan and how active their disease is at presentation. A lot of that's integrated into determining what initial therapy should be provided. The stance from the MS Society and from the American Academy of Neurology and worldwide organizations, for example, is that MS patients should be treated with a disease-modifying therapy. We've gone away from saying, watch and wait, this looks benign. We do believe that there is a real reason to have almost everybody with the diagnosis of MS at diagnosis be treated with a disease-modifying therapy. The choice then becomes, again, a very practical one. Can we get this person a drug? Are they able to take it, a shot? Maybe they're averse to injections. Are they allergic to some form or a compound that could be important? Do they have access to an infusion center, et cetera? And really, side effect profile. We go through very carefully. This immune modulatory therapy has a degree of immunosuppression that could put you at risk. And during the pandemic, of course, that was a major concern for many, many patients. So it really is a compilation of many different and some nuanced variables that we discuss with the patient. Again, I don't want to really skew any perceptions here, but at our institution, we do heavily rely on B-cell depletion therapy. That's one of the reasons I chose ocrelizumab as an example. This is a very highly effective therapy. And many of us were debating even just a few years ago whether or not we needed to use a highly effective therapy up front, or should we reserve that for later? In my view, and this is just a personal opinion, it's a little bit like time is brain, or time is, why waste any opportunity? If I had cancer, I don't want the first level therapy that could treat me very effectively. I want to go to the most effective thing. So many of us have kind of evolved into that. When I say us, I mean even outside of academics. We're going to use a very potent therapy like ocrelizumab that can get us a very good chance of controlling disease early on. I hope that somewhat touches on that very complicated and difficult question.
Yes, thank you so much. However, if I could just do the follow-on question, then what in your experience has been the most troublesome or the most frequently occurring adverse side effect when using ocrelizumab?
Oh, so ocrelizumab, it's probably the increase in infections. And we're not talking about the crazy ones. And I was going to answer your question if it wasn't specific about ocrelizumab, what's the most troublesome consequence of all of our therapies altogether? Everyone's got their eye on progressive multifocal leukoencephalopathy, or PML. We haven't seen that to any sort of measurable degree in ocrelizumab. That came primarily from the other therapy I mentioned or alluded to earlier, natalizumab. Ocrelizumab has that immunosuppressive quality because you're missing B cells. Some or most of our patients then drop their immunoglobulin levels, and many people have a tendency to have more basic infections, upper respiratory things, urinary tract infections. So not severe, not like PML, but day to day, it does somewhat trouble patients to be plagued with these other, common infections that are more frequent now and can be more serious then.
So in other words, those associated with general suppression of the immune system. Would that be fair to say?
That's fair.
Okay. All right. Thank you so much, everybody. Unless there are any other questions for Dr. Wu. Oh, I'm sorry, Dr. Strick, please.
Yeah, no worries. No. I was just wondering, I don't know much about stem cell therapy, but what is your feeling? Yeah, what's your feeling about it? What do you think will happen in the future?
Yeah, I think it's super exciting. And I do think there's a lot of promise. The trial that I highlighted, Beat MS, has made very strict parameters as part of the inclusion-exclusion criteria, because I do think what we'll end up using that for in the future will be highly active patients, young, relatively healthy individuals with very little disability that will eventually have very severe disability. We will be able to spare them, I think, with bone marrow transplantation. Unfortunately, a majority of the individuals volunteering for this trial are those with long-standing MS who probably are, experiencing a progressive form because the disease has affected them so severely. And I do not think that bone marrow transplantation in this form will be very helpful for that. So what do I see as maybe the future for it? Really offering an opportunity to be disease modifying and pharmaceutical free after undergoing an autologous hematopoietic stem cell treatment early in the disease course.
Yeah. Fingers crossed.
Fingers crossed.
Yeah, thanks.